Good day, everyone. Before we start our lesson, I flashed these pictures in the slide. So I think everyone are quite familiar with these pictures, right? But what's something or a word that could represent all of this picture? Or one word that is related to all of these pictures? Do you have any idea? I think everyone has idea already since these are quite familiar to us now. So we have here a man flexing his bicep, an egg or a boiled egg, some foods like salmon, cheese, beans, chicken. We also have a girl with unhealthy hair, a whey protein, a girl with a healthy skin or a glowing skin, and a cat. So later, we will know if your guesses are right. So today I'll be discussing about protein. So protein is quite familiar to us. I think everyone had already heard this word. It's not something stranger to us, right? It's very familiar. For the objectives of our discussion, at the end of the lesson, the learners are expected to know the definition of protein, to learn the structures of protein, and to understand the importance of protein. During our previous discussions in biochemistry, we had learned that there are biomolecules that our body needed. These are carbohydrates, lipids, nucleic acid, and of course, protein. My task today is to elaborate and help you to understand what is protein, its structure, and its importance. Protein is a biomolecule that is made up of amino acids. Then it is joined by peptide bonds. Later, we will know what is a peptide bond, and it is considered as the building blocks of life. And later on, we would further discuss why is protein considered as a building blocks and its importance to us. Protein are produced by living organisms, and most of them are organic molecules. We always hear that protein is important. We need protein. We should intake protein. So these are the some of the importance or common importance of protein. Building and repairing tissues. Proteins are the fundamental building blocks of cells, tissues, and organs. They are essential for repairing damaged cells and tissues. 2. Supporting the immune system. Proteins help produce antibodies, which are pivotal for fighting off infections and illnesses. And maintaining metabolic function. Proteins are involved in numerous metabolic reactions, including those that break down food, convert into energy, and eliminate waste products. They also help in regulating fluid and acid-base balance in the body. Building and maintaining muscle. Protein is essential for building and maintaining muscle mass, which is important for strength, mobility, and metabolism. Aside from that, protein also help in supporting bone health. Protein helps produce collagen, a key protein in bones, contributing to their strength and structure. It also helps in supporting the body's ability to retain calcium, which is pivotal for strong bones. Another thing is that protein helps in promoting satiety and weight management. Protein can help you full and satisfied after eating, which can aid in weight management. If you can notice, athletes and bodybuilders eat more proteins. They are into eating protein bars, taking protein supplements, drinking protein shakes, or prefer eating chicken breast, salmon, and eggs because it can make them full faster and will not add a significant value on their weight. And did you know, guys, that cat contain more protein than chicken breast? Well, it's been a me circulating online.
Protein can also be an energy source in some cases or situations where body cannot utilize carbohydrates or fats. Protein can be a source of energy.
Protein can be an essential for growth and development as well. Protein is crucial for growth and development of children, teens, and pregnant women. Also, protein helps in nutrient transport and plays a role in clotting and vision. So by those importance were enumerated earlier, we can really assess that or understand that protein is really essential for our body, not just for building muscles, but also in helping in the growth and development, in bone health, promoting satiety, weight management, supporting the immune system, maintaining metabolic function. It can also be an energy source as well. So in a nutshell, we really need protein in our bodies. We have here protein structure and function. Proteins, as mentioned a while ago, are essential bioorganic molecules with diverse importance, roles, or functions in the body. About 100,000 different proteins exist in humans, which are pivotal or vital for enzymes, hormones, and tissue repair. Another notably mentioned a while ago is that protein is made of amino acids. Amino acid is the monomer of protein. There are three functional groups present in amino acid. One is the amino group or the amide. Next, the carboxyl group or the carboxylic acid. And the R group or the side chains of the amino acid. Amino acids will always have an amino group and a carboxyl group and an R group. However, it will vary in terms of size, charge, and reactivity. Always remember, only L amino acids are found in the proteins. As you can see, L amino acid has the amino group on the right side, while the side chain or the R group is on the left side. To visualize it and understand more, here are some examples of amino acids. First, we have leucine and serine. So leucine and serine both have amino group and a carboxyl group. So we have here, they also have the R groups. R groups are present in both amino acids. However, they are quite different. Leucine has a nonpolar alkyl group, which is isobutyl, and serine has a polar hydroxymethyl group. Another example we have here, valine and histidine. Histidine features an imidazoles ring that is aromatic and potentially positively charged, while valine has a nonpolar isopropyl group that is hydrophobic. In the situation of alanine and glycine, they differ because alanine has a methyl group as its side chain, which is a nonpolar, while glycine has a single hydrogen atom as its side chain, making it the simplest amino acid with the most flexibility since it only has a hydrogen atom as its side chain. And these are the 20 common amino acids. We have alanine, glycine, isoleucine, leucine, proline, and valine. We have also phenylalanine, tryptophan, tyrosine, aspartic acid, glutamic acid, arginine, histidine, lysine, serine, threonine, cytine, methionine, asparagine, and glutamine.
The acid-base properties of amino acids. Amino acids can exist as torians, which means both positive and negatively charged. It also have exoelectric point. At this pH, amino acids have no net charge and do not migrate in electric fields. Peptide bonds and chains. Peptide bond formed by dehydration between carboxyl and amino groups. 2. Directionality. Chains have N-terminal and C-terminal ends. Backbone. Repeating peptide bonds and alpha carbon form the backbone. These are the peptide forms. If 2 amino acids, that is called dipeptide. If 3 amino acids, that is tripeptide. Short chain of amino acid, typically less than 20, is called oligopeptide. Now, if there is a longer chain of amino acid, typically greater than 50, and that is polypeptide. This is an example of peptide. This is a compound named aspartame. It has aspartic acid and phenylalanine as 2 amino acids, so it is a dipeptide. Compound glutathione, and it has a reduced form of glutamic acid, cytine, and glycine, and this compound is an example of tripeptide. The hormone oxytocin is an example of oligopeptide. There you can see there are many amino acids present in oxytocin. An example of polypeptide is hemoglobin. And insulin is also a concrete example of polypeptide. There you can see there are so many amino acids in insulin. Small peptides and their functions. Peptides can act as neurotransmitters, like enkephalins are pentapeptides that reduce pain. Also, it can be an antioxidant. Glutathione is a tripeptide antioxidant in cells. Peptide versus protein. Let's differentiate peptide from protein. Peptide is a short chains of amino acid. Also, specific biological functions like signaling, hormone regulation. Examples are insulin and oxytocin. While protein, on the other hand, have longer chains of amino acid. It has complex 3D structures, diverse biological function, example enzymatic activity, structural support, and immune response. And examples are enzymes, antibodies, and collagen. It is important to know that proteins differ from each other through amino acid composition, sequence, location, function, and spatial configuration.
Protein structural characteristic: simple versus conjugated protein. Simple proteins made only of amino acid, while conjugated proteins with additional chemical compounds called phosphatic groups. We have here the types of conjugated proteins: hemoproteins, lipoproteins, glycoproteins, phosphoproteins, nucleoproteins, metalloproteins. Hemoproteins contain a hemo group like hemoglobins. Lipoproteins has fats, and glycoproteins is a carbohydrate group. Phosphoproteins contain a phosphate group. Nucleoprotein contain nucleic acid, and the example of nucleic acid is chromosomes. Hehaloprotein contain metal ions. Example is the iron, zinc. We have here the different types protein structure. The first one is the primary protein structure. The amino acid sequence in the protein chain define its primary structure. Similar structure exists across species like insulin in cows, pigs, and humans. Linear sequence of amino acid in a polypeptide chain. Sequence order determine protein's identity and function. To determine the sequence, we have here the N-man degradation short peptides has amino acid one at a time from the N terminal. The second one is the mass spectrometry, a modern fat and used for full sequence analysis. Species similarity proteins sequence can be conserved along animals. Secondary protein structure, alpha highlights. Right-handed spiral formed by the hydrogen bonds between every 4 amino acid. It's a coil stabilized by hydrogen bonds every residues. Beta-plated sheet is a strands lay side by side forming a sheet held by hydrogen bonds. Strands aligned side by side can be parallel or anti-parallel. Tertiary protein structure. From the word tertiary, it's a tree. A full 3D shape of a protein resulting from the folding of its secondary structure, active site, and biological function. Stabilizing interaction. Disulfide bond is a strong covalent bond between sulfur atom of cytine residues. Hydrogen bonds between polar side chain. Hydrophilic interaction, a polar group cluster inside the protein away from water. Salt bridges is an ionic bond between positively and negatively charged side chain. The fourth one is quadratary protein structure. These structures are not always present. The arrangement of multiple polypeptide chain or subunits in a protein. For example is the hemoglobin. Hemoglobin has 4 subunits. To identify the quadratary protein structure, it has a multiple subunit, specific arrangement, interaction between subunits. So consistent of 2 or more polypeptide chain. Subunits are arranged in a specific 3D structure and held together by hydrogen bonds, ionic bond, hydrophobic interaction. So we have here the primary structure to quadratary structure. The primary structure has an amino acid sequence. Simple lang siya. While the secondary structure has a alpha hexase and then it has beta sheet, plated sheet. It sequence folds into 3D structure. Moadto na din ta sa tertiary structure. Matured protein holds onto itself. Then goes na din siya sa quaternary structure. Protein consists of multiple polypeptides chain. Protein hydrolysis and denaturation. Hydrolysis, it means splitting with water. It's a chemical reaction that breaks peptide bonds, releasing individual amino acid. Ang process niya is to require water and often enzyme, like pepsin and trypsin, or strong acid-based covalent proteins into amino acid that can be absorbed or used by the body. While the denaturation, unfolding or misunfolding of proteins losing its natural shape and function without breaking the primary structure. causes by heat, for example, cooking eggs, pH changes. Example is stomach acid. Chemicals, for example, is alcohol or urea. So for the summarization of protein structure, the primary protein structure is a sequence of amino acid. The type of bond is peptide bonds. The secondary protein structure is a local folding of alpha hexes and beta sheet, a type bond of hydrogen bonds. The tertiary protein structure is a 3D structure of whole chain, and the type of bond is hydrogen bond, ionic bond, disulfide bond, and hydrophobic bond. And the type and the last protein structure is the quaternary protein structure. It's a multiple subunit arrangement. Ang type of bond niya is same sa tertiary protein structure. Sa ganito, hydrogen bond, ionic bond, disulfide bond, hydrophobic bond. And for the summary of hydrolysis is to break peptide bond into amino acid. It uses water or enzymes. And denaturation is a loss of structure or function. It uses the drops of 2 degree to 4 degree except sa 1 degree.